arguing who gets to introduce Dr. White, and I'm just going to say thank you, Dr. White, for coming and joining us. And I'll let the host take it away. You guys have a great show.
Thanks, Ken.
Thank you, Ken.
Av, go ahead. You take the honors. Oh, all right. Welcome, Dr. James White. I'm really excited to talk to you again. It's been a couple minutes since we last caught up, and dying to hear what you're working on these days. So please, without further ado, please shed the light.
Okay. Well, we are in my lab. We have a number of projects that are ongoing, and of course, we're always interested in Rhizophagy cycle and how that process works, and measuring nutrients that Rhizophagy cycle brings to plants and that microbes bring to plants. So we're doing that, but we're also looking at how plants actually guide the bacteria into the root tips. And we know there's nutrients there, but we think there also are charges. We can see evidence that there's charges on the tips of roots, and it seems like it is a negative charge on the root tips. And we think that bacteria have a positive charge when they have their nutrients. So there's kind of an electrostatic attraction, right? Electric attractions. This is something in an area that people don't know very much about in terms of how plants work and how plants use these charges. But they actually are quite charged. You've heard about the people use a potato for a battery and stuff like that. So there are these charges in plants, but that may play a role there. The other thing, another thing we're working on is we're trying to develop a bioherbicide that's totally edible, and in fact that uses the plant's microbiome to kill weeds. And we're developing. We have prototype now with the U.S. Geological Survey as a collaborator, but this herbicide that we're experimental herbicide that we're working on, bioherbicide, will use amino acids to turn bacterial symbionts into pathogens and kill the plant. So it's manipulating these normally beneficial endophytes in plants to turn them into pathogenic or harmful to the plant. So, but that's ongoing. The other thing that we have that's really interesting is we're working on nitrogen fixing corn. This corn land races that appear to fix nitrogen, compared to commercial cultivars, and that we have a project with Walter Goldstein at the Mandamin Institute in Wisconsin. He's a corn breeder, organic corn breeder, who's been, for the last 10 years, he's been working on this, trying to breed these land races into active, into cultivars that people can use, especially for organic cultivation, so they don't have to put nitrogen on any of their plants. They don't have to keep putting all the other kind of organic fertilizers on plants. And the thing with that's most interesting is the latest finding is that in this organic corn cultivars, these landrace nitrogen fixing cultivars, the bacteria actually go into and around the chloroplasts. And we think that this is a symbiosis in a symbiosis, that in fact there are bacteria that are symbiotic with the chloroplasts in these plants. And we can use our nitrogen histochemic, our stains that we use to detect nitrogen, and we could see actually nitrate forming in these chloroplasts. And the other thing about the chloroplasts is they don't break down. And so these nitrogen fixing cultivars stay green all the time. And the chloroplasts in these other commercial cultivars they'll break down and they lose their chlorophyll. They keep recycling what's in the chlorophyll, what's in the chloroplasts. But these nitrogen fixing cultivars don't. They don't recycle it. They keep their green chlorophyll. Their chloroplasts stay green. Yeah.
Is this the cultivar out of Mexico with the aerial roots covered in slime?
That's what people, yeah, that's, some of these are there. Yeah. Some of them, we have some from Peru. And so some of them are more tropical, right? Some of them are more montane. Some of them are more riverine. They'll occur like along the Amazon River. So they're different ones. And we don't, the ones, in fact, we have both. We have from a tropical one, and we have one from montane, this Mexico types, like the one with the aerial roots, right? But they both seem to be doing the same thing. And it's really interesting, because if you think about corn, in the blades of corn, you have different regions, and some regions are low oxygen right around the vascular tissues in an area called the bundle sheath, and some are high oxygen out in the mesophyll and the leaf blades and in between the vascular tissues, right? And we think that most of the nitrogen gets fixed in these bundle sheath and the chloroplasts in the bundle sheath cells. But there's a lot to do there. It's right now it's a hypothesis that we're trying to develop, and I mean that's those two things are the most exciting: the Phragmites project and the nitrogen fixing corn project. Related to the aerial root thing, people think that the aerial root is that those were, in fact, I thought the same thing 15 years ago, that was a source of nitrogen, and it may be a small source of nitrogen, but corn plants are actually getting nitrogen all over their structure. We think that these chloroplasts might be the more important source, but of course that all has to be proven. We've got some proving to do, right? We only have a hypothesis now. We've got some proving we have to do.
How far along are you in this project?
We've been helping Walter Goldstein for about 3 years now, but this the chloroplast symbiosis idea is very, very new. We just the past couple of weeks, we developed that based on some histochemistry that we did in looking at these leaves compared to some other commercial cultivars. Actually, it's really cool. These other commercial cultivars, the chloroplasts break down. They don't have bacteria in them, or if there is bacteria, there's not much. They don't produce nitrogen in those chloroplasts, and the chloroplasts break down. The plant is recycling these chloroplasts, probably because it doesn't have enough nutrients. But in these nitrogen-fixing ones, the chloroplasts don't break down. We can actually move the bacteria from these nitrogen-fixing corn types into other plants and get nitrogen fixation happening in these other plants, and also preserve the chloroplasts where they don't break down. So the bacteria can protect the chloroplasts in not just corn, but other plants as well. And so those are the experiments that we're doing right now to kind of follow up. You could imagine there's going to be a lot of work to actually prove that hypothesis.
Oh yeah.
You could imagine.
Oh yeah. Yeah, I'm working on a project similar to that. We're getting into metabarcoding, genetics, metabolics, and all kinds of crazy stuff. But in those cultivars now, I think the ones I'm familiar with tend to grow a lot taller and throw a lot more ears, correct?
Yeah, they do. They're but of course, if you were making your own nitrogen, you could do that. Yeah.
So are they throwing like 3s, 4s, and 5 ears on the stalks, or even more than that?
I don't even know, Leighton. I can't tell you. It's the corn breeder who, on the other end, that the plants that we work with here are much smaller. I'm told that these will get up to about 10 feet tall.
Yeah, yeah.
Yeah, yeah, and probably having lots of ears on them, and I just haven't seen them there. I haven't gone to the breeding nursery. The plants that we have right now, I have some in some raised beds that we're working with now, but these are the ones at this stage of development are only about 2 feet tall, something like that.
Okay.
Yeah.
The reason I asked this is I've been working with a farmer, Kyle Moat, who actually came on this podcast to talk about the end results of his harvest, and he was throwing 3s and 4s and even a couple 5s, and the people around him in Illinois didn't believe him. He had all kinds of people coming. It was like a 3-ring circus because they'd never seen that before.
Yeah.
And Av, this is something new too. Kyle recently was forced to till because his cover crop wasn't high enough to roll or crimp. And when he tilled, he came across hundreds and thousands of worms. One application of a biological, and now he's got soil structure. Everything is clotting. It's aggregated now. It's not dust. He used to pick it up and it would just fall through his fingers. So again, the power of stuff that we're unlocking right now is just insane. And James, man, this is just mind blowing that you can pull the genetic or the bacteria from one plant and introduce it to another and make that plant even stronger. And that'll have huge ramifications on all crops.
It really does. I mean, we could use these microbes or go from the wild plants and put them back into our crops where we lost them. You know, some crops may still have them, right? Some, like cannabis, probably still has most of its microbes, but a plant like corn, I mean, the way that we've treated corn over the years with the tissue culture and lots of chemistry on them, we've negated a lot of the symbiosis that they had. That's evident. We can tell these tissue cultured plants they're really, really weak plants, and most of our modern corn has gone through a tissue culture phase. So we probably, if our corn ancestors ever had these nitrogen fixing symbioses, we knocked them out. We certainly knocked them out because we thought they didn't need any of that. We didn't realize they were there, right? We thought all these microbes were just pathogens. We didn't realize that they were important to the plant.
So, and we're definitely talking about a bacteria, not a fungi, correct?
It's definitely bacteria. Bacteria, well, that's an interesting point. Bacteria are the only prokaryotes are the only microbes that are actually able to fix nitrogen. They have nitrogenase genes, okay, but fungi will also internalize bacteria. And so you could have a fungus that has nitrogen fixing bacteria in it. And we see that actually in some fungi. We can stain with these histochemical stains for nitrate, and we can see the bacteria inside some fungal hyphae, and we can actually see nitrogen coming around the bacteria. So it's apparent that fungi are able to do a similar thing, not like Rhizophagy, not exactly where there's a cycle, but they can internalize microbes and can use those bacteria as sources of nutrients. But that's a whole new area, and that also has to be proven, right? I mean, I could say it and say what we see, but that's not proof yet. We need more evidence. I have a graduate student starting this fall, and this graduate student is going to work on that and hopefully gather the proof that we need for that.
So I know that this was happening in the mycorrhizae world, but is it happening in the saprophytic world as well?
Yes. Yeah,
wow.
a lot of the it certainly is. The group, and it's interesting, the group that does it is the Zygomycetes. Okay, so this is a primitive group of land fungi. They don't have any swimming stages, so they're not aquatic, but they're very simple. They tend to be, I mean, they're oftentimes called sugar fungi because they'll grow on strawberries and sometimes they'll grow on feces, stuff with simple nutrients in them, right? Fruits and things like that. They'll grow on fruits, but they're not complicated in terms of they don't have the ability to break down wood or even cellulose, right? They're basically sugar. So we didn't think they did anything. But when we look at them with these histochemical stains, they're full of bacteria, and we can detect the reactive oxygen around them. We can detect nitrogen around them. So it's evident that they're actually appear to be getting some nitrogen and probably other nutrients from bacteria that they internalize from the soil. And they're everywhere. The mycorrhizae are from this same group of zygomycetes, right? The glomalin, the VA mycorrhizae, they're actually diverged off of this zygomycetes group. And they're considered in the greater zygomycetes, they've been considered to be in that group for many, many years now, even though people keep floating them back as a separate group, but they really are in that group.
So this is the Oomycetes, right? I always screw up that pronunciation.
Not the Oomycetes. Those are aquatic. Those are typically aquatic. That's an aquatic group. We don't know if they're doing this or not. The Oomycetes. We don't know. We don't know. We know we can see the bacteria in the Zygomycetes and Ascomycetes and in some Basidiomycetes, but we don't know about the Oomycetes. It's a kind of a separate group, but it's interesting. But you know what this does, Leighton, and this tells us, to me, how little we really know about how these organisms are working. Including plants and fungi. Let's add to algae. Algae. I don't know if you knew the latest one of the latest studies we did on rhizophagy. Is we looked at algae, and as it turns out, roots can actually absorb algae into their root tips, and they degrade the algae with superoxide, with reactive oxygen, get all the nutrients, and we can see when roots are doing this, they will turn green because they absorb the chlorophyll. So you get green roots when they're absorbing algae into their roots. And the other thing that happens is algae also will have their own endophytes and their bacteria that they carry inside them. And so when the plant absorbs the alga from the soil, it gets the nutrients in the alga. Most of that appears to be fully degraded, like chlorella is an example. Chlorella. But the endophytes in the algae aren't fully degraded. Instead, they go out of the alga and they go into the plant cells. So the plants then are able to then get these endophytes from the algae in the soil that they absorb. And that was a quick little study that we did for a company. I'm not mentioning any company names here, but it was a cool outcome. And the rhizophagy cycle can involve more than just bacteria, and even fungi can involve algae, and even possibly blue-green algae. We don't know yet, but it certainly green algae, because that's our evidence that we have for that experiments.
Is penetrating, coming into the cell wall the same way as in the rhizophagy cycle?
Exactly the same way.
Okay.
Exactly the same. Yeah. No, it was really interesting that the process is broadened because these plants can absorb algae from the soil as well, and it is really amazing. It is amazing, and the chlorophyll just goes into the plant cells. And the other thing that happens when they're eating algae, when they're absorbing algae into their root cells, is that besides turning green, the roots, they also appear to have another effect. They appear to be more to absorb more bacteria when they do that, to take in more. We say that it's kind of like humic substances. When plants detect humic substances, what they'll do is they'll start to take in more bacteria. And we think that's a signal to the plant. The humic substances is a signal that there are microbes degrading plant material. Humic substances happen to be the breakdown products of plant material by microbes, right? So when plants can detect that, it's a signal to the plant that there's microbes there that have nutrients from plants, from degrading plants. So they start to take in more of the microbes, and we can see that. Well, the same thing happens when you feed them green algae. They start to take in more bacteria. There is this similar humic substance effect, I think this is why the green or the brown algal extracts work. People have algal extracts that they put. Well, it's probably something in the algal extract, and it may have to do. I mean that we may see the same thing when we give them these green algae.
Are they taking them in as endophytes or taking them in as part of the rhizophagy cycle?
The bacteria that they're being taken in part of the rhizophagy, because they're degraded. They are actually degraded when they go into the roots, into the root cells. They're broken down, and at least most of them appear to be broken down. We can actually see that when the alga goes in, the plant hits them like chlorella, right? Green algae. They hit them with superoxide, and then the first thing that happens is the chlorophyll starts to vanish from the alga. And that may be that the superoxide, the reactive oxygen, is breaking the chlorophyll down. The other thing that happens almost simultaneously is that the bacteria that were in the alga begin to jump out. Jump out. They don't have legs, but they begin to go out of the algal cell, and we can see them forming little halos around the algal cell. And then eventually that algal cell vanishes, but those bacteria stay inside and they're replicated. Yeah, it presents a picture of plants as attracting not only bacteria and yeasts, but also green algae. So the more diversity of life you have in that soil, the more plants are going to be able to get to it. And you get, something like an alga, this is they're autotrophs, right? They're photosynthetic. So they're feeding these microbes. They have their own endophytes. And when a plant swallows them up, absorbs them into the roots, then they get those endophytes. So it's a source of endophytes that are already adapted, possibly even nitrogen fixing endophytes, that are coming from these algae that plants can then take. So it's kind of a different picture of even rhizophagy when you consider algae.
Yeah, I was going to say this is like next level above. So the bacteria are then becoming an endophyte. They're living in the cell. Are they coming out of the cell again and entering back into the rhizophagy cycle?
They are. They're going back into the soil. Yeah.
So are they then reforming algae in the soil?
It's hard to say if the bacteria, they will probably colonize more algae, but they apparently are free living, have a free living capability as well.
Mm.
Wow.
Yeah.
I'm just, my head's spinning now.
I know. Well, things are a lot more complicated than we initially thought. Our initial idea that plants, work by chemistry is, further and further from complete, right?
Yeah, further and further from the truth.
From the truth, yeah.
So
I mean, there are chemicals. We can feed them chemicals, right? But they're more active at getting chemicals from the soil microbes than anyone ever thought.
Yeah, from the sand, silt, clay, and the organic matter. They're pulling everything that you need.
Yeah.
we just got in the way, and as a result, we're all sick now. But anyway, we won't go down that road. Oh man.
Yeah, well, it's true. It is true. It is true. We have damaged ourselves, but that's not proved though, because there some of the studies on like Neanderthals, they had arthritis and they had some issues too. Of course, they also had much more physical lives than we have. So, I mean, they could have gotten arthritis from not just having bad diets, but from how hard they work, wrestling grizzly bears and stuff like that, jumping on mastodons. That would certainly cause some problems.
I mean, there's a couple of things. One,
yeah.
I think the whole nitrogen fixing of within chloroplasts, or in association with chloroplasts, I mean, that's a whole new understanding of nitrogen dynamics in the plant. The idea that we are, they're producing nitrate nitrogen. It's not in an amino acid form, and all that happening right in the leaves is pretty phenomenal. And then I also wanted you to perhaps comment to our audience. I think it was through you I first learned that Fusarium doesn't have to be pathogenic, and in association with other bacteria, it is not a plant pathogen. Would you mind expanding a little bit on both of those comments?
Yeah. So I'll say in the big picture of it that just a generalization. Okay, a couple of things. One thing is that in nature, disease is not a major issue typically, and people have always said, well, it's because there's not a monoculture in nature. It's mixed. Okay, so that's part of it, right? But actually, that's only part of it, because part of it is with the microbes, with bacteria colonizing fungi, it alters their virulence, and that's either because the bacteria that colonize fungi will weaken them. Okay, that's one idea that we can see that. With Fusarium, we think that's happening. But the other idea is that with fungi free living in the soil, they're not getting their nutrients from plants, from degrading plants or causing disease in plants. They instead may be getting a significant amount of nutrients from the symbiosis that they develop with bacteria. And so when they internalize bacteria into their hyphae and get nutrients from them, the fungus, in a sense, is satisfied, right? It has all its nutrients. If we remove those bacteria from the fungus like Fusarium, the virulence goes way up. They start to grow faster, they start to sporulate, and they attack plants. Okay, if you look at seeds, for example, basil, the herb, right? Seed. The seed has a little matrix on it, and there's bacteria all over that seed. If you challenge that with Fusarium, a pathogenic Fusarium, it doesn't. It typically will not have much effect on the seed and seedling. But if we take that seed and we sterilize it, we soak it for a time in Clorox and removes the microbes on the surface on the seed, and then we put the Fusarium there. It eats the seedling up. The seedling doesn't have a chance. Essentially, the bacteria are part of the immune system of plants, and so when you have plants with lots of microbes, they're more protected from disease than plants without microbes. So many of our disease problems that we have in our major crops are likely due to the fact that we've removed many of the microbes from those crops. And so that's it. That's the relationship right there. If the fungus, the hypothesis I like the most, but of course needs more development, is that that fungi are getting nutrients from the bacteria, and that if they can get their nutrients from bacteria, then they don't need to attack plants to get their nutrients. But we need, that may not apply to all fungi. We have some fungi that are obligate biotrophs, that things like rusts, right? Rusts cannot live in the soil free living, or anyway free living. They can only live as parasites of plants, and so that may not apply. They may have adapted. There's some reason to think that rusts and some of the obligate plant biotrophs don't interact with bacteria as much as some of these other free living fungi. Fungi that have both a free living phase and a parasitic phase.
You had already mentioned something regarding tissue culture and that perhaps leading to some of the lack of microbial response with corn to whether it's a biological pesticide or microbial inoculants. Cannabis is definitely moving towards a lot more tissue culture. There's a lot of interest in especially with hop latent viroid. What would your recommendation be if you were a cannabis grower and you received tissue culture plantlets? What would your first step be in terms of trying to get that health back into the plant?
Well, it's a good question. Because people are going to do this, we're going to play with our crops. We're going to do, tissue culture is an art now. It's an art and a science, right? But it's a high art. In fact, I have a well-known tissue culturist here in the lab near me. And the plants that she's worked on, if I look at them microscopically, I can see they're really depauperate of microbes, and they don't develop the same without, they require, you have to put fertilizer on them and all kinds of stuff. They become dependent on fertilizer. The way that you could put microbes back in these tissue cultured plants would be to grind up, if you have seeds, especially seeds, grind up wild plants or other cultivars that haven't been tissue cultured, grind the seeds up, or take the seeds, germinate them, and then grind up the tissues of the plant, get some juice out of them, right? Get the juice out of them, and then take that and put that on your tissue cultured seed and let that germinate in that juice, and then they'll absorb those microbes from the juice. Yeah, no, that's how we. So we have that's how we do it here. You don't have to isolate. In fact, it's better not to try to isolate the bacteria, because you don't know what bacteria are involved. There may be multiple bacteria, and you may not even be able to cultivate it. It's like hit or miss if you're trying to isolate. It's better, and I should mention also, the technique that I talked about is very similar to the technique that the American indigenous used to use to get microbes in their corn, back in their corn. And they would, they had been cultivating corn for a long time. They had something called a corn medicine. And what they would do is they would go out and get wild grasses and dig them up in the woodlands, get the roots and put them in water and beat the roots up, warm the water a little bit to reduce some of the microbes that were in there, and activate endospores like Bacillus endospores. And then they would germinate their corn in it. When the roots came out a little bit, just poked out of the corn kernel, then they would go take that and plant those in their mounds with their other two sisters for their three sisters agriculture, or their fields if they were doing fields. That's a published technique that they used. There was a Iroquois, there was a holy man actually that wrote Iroquois named Handsome Lake was his name, but he wrote about that corn medicine technique.
Oh man, so many questions. All right, let's reel it back in a little. The fungi, when it takes in a bacteria, is it going to consume that bacteria, or is it going to also perhaps just take off the outer shell, steal the nutrients, and let it go back out, similar to the rhizophagy cycle? Do you know?
Yeah, some are fully consumed because we can see them breaking down. Some bacteria will be fully degraded, and we can see that inside root cells. We can see them vanishing into nothing. Swelling, what happens is they'll lose their cell wall and they swell a little bit. Some pseudomonads swell a little bit and eventually just disappear in there. Others, but they're also replicating, so some of those will go all the way out. So, and we do know that the what determines what happens to the microbe is how resistant it is to superoxide. And those that are highly resistant don't break down at all, and they become a problem for the plant. Those that are highly susceptible likely break down all the way, highly susceptible to reactive oxygen, to superoxide. They'll break down all the way. There's a sweet spot right in the middle, and those are the ones that will lose their cell wall. Some of the cells will break down, but there will be enough surviving that then they will go into the root hairs, and as those root hairs grow, they'll eject them out of the tips of the root hairs. So there's variability. The worst situation is when the microbes are too resistant to reactive oxygen. Because if they're too resistant, the plant can't control them. And so they go in and they become inhibitory to the plant. But mostly that's not a problem because plants have their microbes, and they, for some way, they exclude many of those that are very highly resistant. We don't exactly know how that happens, but when we put them in the plant, the plant, if we remove all the microbes and put them in the plant, then they inhibit the plant. But when the plant has its microbes and we put these resistant ones there, there's no effect on the plant. Somehow they exclude them. We don't exactly know how they exclude them.
So it's kind of like quorum sensing. Like they know that this is one that they don't want to necessarily pull up.
People say that, Leighton. They mention is there a quorum sensing, and I don't. If there is a quorum sensing, I would think. I mean, it can be inside the plant cells even. I can't say. It's sort of hard to. It's hard to know. The root tips can take in one microbe at a time, or they could take in many. But you might be right. I mean, you might be right about that. Maybe if there's not enough of them there because all these others are there on the root tip, that would essentially not have a quorum enough so that they can then be absorbed, right? That's what you're getting at, I think, Leighton. And that could be, yeah, it could be there. That could be that plays a role, but I just don't know for sure what the mechanism is.
So let's talk for a minute about border cells. So border cells are basically the guards, right? That prevent the plant from either anaerobic conditions. They release themselves and the root pulls back. But are they also the ones that are opening the doors to these pathways that are letting cells in and out of the root tips?
So when you say border cells, you're talking about the root cap cells that are sloughed off, right? Yeah, those are the cells that are sloughed off. There's the classic theory of guard cells is that they are a protective layer, kind of a kamikaze layer on roots, so that if the root hits a rock or something, that it won't hurt the meristem, right? Underneath these meristem is growing, and that's more important. So these are kind of these root cap cells are protective. That's the theory, the classic idea about these root cap cells. But what we're seeing with these root cap cells is that they are sloughed off and they're full of microbes, and they go along the root column as the root's growing, and they're depositing them right along the column. So they're essentially, we've heard it may be another way that the plant is putting these good microbes right outside the root, in that root column. Besides shooting them out through the tips of the root hairs, as those root hairs grow, depositing those microbes. The other thing is we can see actually nitrogen being fixed in some of those root cap cells. So it may be another way to put nitrogen right around that root.
Well, it kind of makes sense that they're using them both as a protective coating as well as an aftermarket defense around.
It does. And people, that's why I never deny some of the old ideas, because they were logical.
It's another good reason to not necessarily remove your root balls if you're using a living soil bed system, and as long as they're disease free, kind of makes a lot of sense to leave all that there.
Yeah. Something about these nitrogen fixing plants, at least this corn, that compared to commercial cultivars, is they're really hardy. They tend to have thicker leaves. They stay green. I have dug up this nitrogen fixing corn and dug up commercial controls several times, replanted, and every time the commercial cultivar dies and the nitrogen fixing line continues to live, I plant it right back. I put it right back, pull it out with the root ball, pull it out with the root and soil there, and then have it out for a day while I'm working on it in the lab. Then I take it back, plant it in the same spot, and the next day it's perfectly good. It's still growing. Those plants are really stress hardy. So one of the factors with having endophytes is what we've seen in a lot of other systems, is this stress hardiness. The plants are just tougher. So you think about climate change and what's going to happen to many of our modern cultivars, when things get hot and we don't have enough water to put on them. Well, a lot of our crops are just going to fail. We're going to have some of these endophyte enhanced, some of these crops that are full of microbes, just because they're going to be much hardier, and that may be the only crops we're going to be able to grow in the future when things get really bad.
So, not to bounce back, but I think it's important for the audience, especially for Av as part of his cannabis working or teaching, was that you talked about making this concoction for you to put your seed in to then pull the endophytes back in. Do you believe it would be true if you used that concoction as a foliar spray, that you could actually get it back into a plant that was already growing?
Yeah. Oh, yeah. Oh, no doubt about it.
Wow.
What happens is the bacteria will colonize the meristems, the shoot meristem, and those meristems, when they're growing, when the leaves are a little tiny, right? They actually have some bacteria growing around them and in them, and that's where there's sugars and stuff around that meristematic area, and so you could put those microbes as a foliar and get them to colonize the shoot. So in theory, you could have like a plant that has microbes in the top part and below has fewer, right? So you could have these, for any given crop, you'd have to test it. Most of the work where you see this foliar working are monocots, because monocots will have, like corn, for example, or vanilla. They have a shoot. They have where the leaves go around it like that. It's almost like a vase, and then they'll have water growing, liquid that has sugars in it, growing in that vase, right? And the microbes are cultivated in that little vase area, that little moist area. So if you ever check corn, if you go in the morning and look at your corn, and look right down into the shoot, you'll see a droplet of water there. And then in the afternoon, it'll be gone, right? It'll be absorbed back in. But you have that constant. The plant is bathing that meristem. And so if you put your microbes there, then they'll be absorbed into those tissues. So yeah, you could absolutely, you could put them right on the as a foliar.
So do you think that pool of liquid, which is probably easy water, is actually a sponge to absorb airborne microbes as well?
I don't discount anything. Yeah, it would likely, and we know that dust particles and microbes float in the air, and raindrops will form around particles, sometimes around microbial bacterial particles. So yeah, I do think that that's a source for other microbes to get into the plant. Maybe even carried by insects, even. That could go for a drink of water and deposit some microbes when they go there. Yeah, I don't discount any of that.
Well, nature is just amazing. I mean, the fact that these cultivars that have the capacity, because of all of their nitrogen fixers or other nutrient cyclers that we haven't even gotten to, that you can pull it out of the ground, leave it in a hostile environment, and then put it back in the ground and it takes off. I mean, what a great evolutionary strategy for resets. So whether it's a flood or whether it's a earthquake or whatever, they can just deal with it and come back, which is just amazing.
It really is. It really is. And if we had perfect knowledge about how plants were cultivating microbes, we would have probably been much more careful in how we cultivated our crops.
Or maybe more advanced.
Yeah.
As a species, right?
Yeah. Well, we certainly, the health issues that we have, in terms of microbiome health and some of the allergies and other things that people think may have to do with the microbiome, maybe we wouldn't have that if we didn't have the idea that we got to kill all our microbes.
Well, we used to treat food as medicine, and that kind of went out the window. But yeah, I mean, it's crazy to think where we could have evolved to if we had been a little bit less power driven and more driven toward the social responsibility and evolution of our own species. Yeah, there could have been a wonderful things that happened, and maybe there's still time. Maybe the sciences will surpass the power and money system to allow us these things that are so important.
I certainly hope there's time. I think there's time. Little by little, it looks like the world is changing. It's
Agreed.
little by little. It's definitely little by little. But I mean, I can see things changing, and people, certainly with how we grow crops, people realizing that what we're doing isn't sustainable, more and more, and people are coming around. It's not instant, but we had a first couple of weeks ago. I never thought it would happen, certainly, but the Gates Foundation had an symposium on endophytes so that they could decide if they should invest in microbial endophytes in crops, if that was a solution, if that was at least part of a solution. So a bunch of endophyte people, and including me, and from all over the world, they invited, and they paid the way there for everybody, and then we had a 2-day symposium, and I hope that the result of that is that the Gates Foundation will begin to invest, and if they start to invest, then I hope the National Science Foundation and the more USDA investment, the more is going to happen. I'm hopeful. So fingers crossed on that.
Those two, right? Everything I can cross, brother.
Yeah.
I mean, you are the potential future to providing food in a ever changing world. I mean, we're never going to stop climate change or migration. I mean, look at the Savannah or Sahara Desert. That was all grassland.
Oh yeah. Yeah.
We have to be able to adjust, similar to that plant that you ripped out of the ground, brought it down for a day, and then let me go.
Take it back in. That's amazing, though, when you think about it. I mean, you rip the plant out. How many plants can do that?
Right?
Yeah. Oh, yeah. Yeah.
Well, listen, we're down to 10 minutes. So, Av, if there's something else you want to ask him.
Just trying to follow up on that, Jim, do you feel like you've influenced any breeders to fully understand how important it is to be cultivating and breeding in a micro dense soil, so that the plant is ready to respond, whether it's to biopesticides that we want to be using or microbial inoculants or a living soil? Have you had any influence there, and have people gotten back to you and thank you for really popularizing this work?
Well, over the past few years, there have been a few breeders who saw this as something important, but typically it was in a proposal writing stage, right? And if the proposal didn't come through, then they didn't follow up. Okay, but this organic corn breeder, Walter Goldstein at the Mandamin Institute in Wisconsin, he's been trying to breed nitrogen fixing corn for the past 10 years. So he's already there. And I think that my feeling is that and this is one reason why I was trying to work with Walter now to try to help him do this, is if we can be successful, if we could put all the information, show how these systems work, begin to develop some actual cultivars that are going to be practical out there, then that's going to prove to the rest of society that we can now do this, that we can create cultivars that are endophyte enhanced, and that it's going to be better, and we'll know how to do it, right? We'll understand what the microbiologist and the botanical part has to do, and what the breeders have to do to maintain those systems, right? So yeah, working together is key. Absolutely right. And if we do it, I think it'll be really important. It'll be a kind of proof, right, that we can do it, and that it's a way and more people will then, I think, begin to do it.
And I think, that's one of the drawbacks, that if we're not breeding in this situations, plants are evolving to not associate with endophytes, not associate with other microbes. And so if we're using things like microbial inoculants, they're going to be like, why bother? Or biopesticides just aren't going to have the same efficacy because the plants aren't used to relating to them. So,
well, we know, for example, that there was a recent paper. I don't have it with me, so I don't remember the details of it. But where they found that some corn varieties can no longer associate with. They've lost the genes, the NOD gene, or genes that are required for mycorrhizal association. So they no longer can associate with VA mycorrhizae. And that's because, we've used so much nitrogen on them and selected them in such a way that they don't need mycorrhizae to survive. They have us squirting nitrogen on them.
Yeah, they got crack.
Yeah. So they're on crack. Exactly right. Nitrogen crack. Exactly right. They're addicted to nitrogen crack.
So are any of these corn cultivars available for purchase at this point in time?
I don't think so. Because the breeding is very much ongoing. I don't think they're testing them. They have lines that they're testing. Some testing in Africa, some testing in other places. I think there's a tropical one. There's one place in Africa. Maybe link up with some more African testing, and also some tropics, somewhere in Puerto Rico, I think, where there's another nursery to test these varieties. So, that's how they do it, testing nurseries, testing areas all over the world. And so it's in that stage of development.
Are they all open pollinated?
They are inbreeds, but I think they will. I think there's outcrossing that can be done. But these are inbreeds, and Walter told me he's trying to put a gene in them that will prevent them from taking in some of the GMO corn, because the organic growers don't want. So they're trying to cross this gene so that it won't outcross, right? Won't take in some of this, because they don't want the GMO genes going into the organic corn.
Wow.
Yeah.
Well, that just makes all the sense in the world to not release it, because you're going to get a GMO cross. Whereas if you get it to not accept that GMO pollen, then you don't have to worry about that cross.
Yeah. Right.
Damn. Wow, dude, you always blow my mind. And it's not fair that we only got 3 more minutes. So we'll definitely have to have you on again. Ken, I hope you got some of the contacts that he was talking about, because I think they would be, great guests to bring on as well, so that we can continue this conversation. I mean, that's what we're here about. The whole game is just grow. And if you don't grow plants, grow internally, grow spiritually. So, a big thank you, James, for taking this much time out of your day and the work that you're doing to provide us with great insight.
You guys are so welcome. I'm happy to do it.
Okay, so how soon can I book you for the next session? I mean, come on.
That's why we have Ken in charge.
We'll give it a few months, right?
Yeah.
Give it a few months.
Awesome.
Ken's been busy. I've never seen a show where he's fired off more gold bars than this one. So thanks so much, Jim.
Oh, you're so welcome. You guys have a good night.
All right. Thank you, sir.
Okay.
Thank
Bye-bye.
you.
Wow.
That was amazing.
Wow.
Yeah. So we do have some questions I think that you guys should be able to help with. The tissue culture based corn are weaker plants. What about cannabis from tissue culture? Just a cautionary tale.
It's the same thing. And he gave you the way to fix it, right?
Mm-hmm.
He gave you the answers to fixing it. So, not only did he identify a problem, he solved it for the community. So,
and the only caveat that I would put to that is that, and of course, I don't know how many seeds he's talking about, but what I would be recommending is that you test your seeds to make sure that they're free of hop latent, and then be able to make that concoction.
Mm-hmm.
Sense?
Yeah. No, that only makes sense. I mean, it's all about the testing. But if you've got the scrubbed ones, and you've got one or a plant that has gone to seed that does not show that viroid, then you're golden. So it's really not like a super expensive or problematic fix. It's really easy.
Well, and we were actually talking about this on the OG's Power Hour this morning, about the generation to generational transfer of the hops latent through actually endophytes.
Oh,
because that's something that Mr. Toad has been looking at, and it looks like it's coming through the endophytes as well.
Which only makes sense. I mean, viroids do work in that manner. They're getting inside the cell out of the bacteria. So why wouldn't they come in through endophyte?
Yeah. But he was talking about, knocking back the fungal side of it, and I think they're attached on the fungal side more than on the bacterial. But there's going to be a lot more research that's going to have to happen.
Well, as he just stated, the fact that the saprophytes and the mycorrhizae are letting these bacteria into them to consume them. Now their cytoplasm has that viroid. So that can come out and get into either another bacteria or directly into the plant itself, in the case of the mycorrhizae. So obviously and we know there's billions of viroids all around us. And so that's part of this whole process. It's once in a while you get an outlier like the hop latent that you have real issue with. And that's to say that it wasn't made because there was a group in the 70s working out of Los Alamos. And I know this because I know the guy that was working down there. And he said they were working on that like they were the coronavirus in 1965. So this is very potentially, because they were trying to get control of cannabis. Did not want us to be enlightened and to be free thinking. Anyway, that's conspiracy theory shit, which we probably shouldn't be talking about. But anyway, what was the other question? Because clearly we lost Av.
Does labs help with the hops latent viroid? I don't believe it does.
No,
but I'm not sure.
Labs. I can't see how they could penetrate the cell and actually dissolve or attack the viroid. You have to get rid of it and then prevent it from getting it. So I'm sorry to say, no, I don't think there's any quick fix on that one.
Oh, don't we wish?
Right?
Yeah. Well, because somebody was mentioning it's actually starting to be part of the seed as well. So that's where you're going to have to test the seed to see if the seed has the hops latent. Then
I don't
want to make that solution.
Yeah, I don't know if we have the technology to test seeds. I know we can do it on the plant, but that's out of my box. I mean, who's the breeder? Dark Horse, I think, was the one up in NorCal that originally came out with that they could actually cure this disease. It was a breeder, a very famous breeder.
That they could actually beat hops latent.
Yeah, you could send them your seeds, and they said they could scrub them, but they didn't take off. So obviously they thought they had it, but maybe they didn't have it, because otherwise it would only make sense for them to release cultivars that were clean, right? Yeah. And so if they were, and I don't know any, people that know them specifically. Like I knew a woman who had called me years ago from Vermont. Her favorite cultivar, which she had been traveling with her whole life, and she was older, had reached out to them. Dark Horse, I think, or Black Horse Genetics. And they basically, were song and dancing her for a period of time. And it basically ended up that they stopped returning her phone calls because she wanted to have that cultivar scrubbed, which they said they could do so that she could keep that cultivar that was so dear to her. I've lost touch with her since then. But obviously, look, if they had gotten it, we don't know about it, right?
Yeah.
Well, thanks for jumping back on, Av. We lost you there for a minute.
Man, it's a hard go. StreamYard doesn't work well for me out here, and I didn't prepare well enough to have my phone charged. So,
a little hint: Starlink is actually on sale right now.
I was going to say that.
Yeah, I got one. Okay.
I have a hard enough time with Bill Gates, let alone.
That was pretty scary. I mean,
it's like what he wants to buy it so that he can control it, or what?
Yeah, well, I mean, there's enough people globally that are studying this that his interest is only fanning us to take more notice of it. So it's not something like the carburetor that ran 200 miles that whoever brought up and shelved. I think it's too late for shelving. I think it's more about acknowledging, oh, fuck, we better at least admit that this stuff works, or we're going to be, run out of town with pitchforks and torches. Literally.
Oh, no comment on that.
All our guns will have been taken away.
Oh, that's amazing. So I don't know if you guys want to end early or you want to carry on just talking about what we talked about with Dr. White.
I'll leave it up to you, Av. I had a very busy weekend this morning, so I'm pretty proud.
As long as our audience members have feel like they've had their questions answered. I mean, we jumped right in with Jim. There was very little background around all of his previous work and stuff. So if there are any other questions or comments, happy to stick around for that. And then
Well, we've got nothing on the back end unless somebody throws something in right away. Do you want to just, you guys were both at Grow Up. Do you want to just mention the significant advantage of having something like Grow Up, bringing the people together?
Yeah, you know, it's like in any of these conferences, all of the best information happens, you know, out in the smoking area, right? You're having, you know, folks just getting together and just chatting, and you're getting experts who are just using salt nutrients, right? And then you've got some who are living soil, and they're having discussions about osmotic potential in the roots, and it's phenomenal. So some amazing dynamic conversations. Been really happy to have all that people, you know, so many different people producing fire in so many different ways. But having a good time having those type of discussions, and that's really, I think it's a testament to the subculture, and a testament to the plant. So yeah, thanks for anyone who did attend, and thanks Leighton and Ken, both of you for coming up to Grow Up. Yeah, Leighton.
I think you did a great job, you and Randy, putting that all together, and kudos to you, because you know that was something I learned early on, is that the best way to teach or learn is person to person, not via this. You can't see my eyes, you can't see my wheels spinning in my head this way, but when you're face to face, you most definitely can tell that, you know, okay, you're either getting it or you're not getting it. So maybe I got to reel it back, or I got to go forward quicker because I'm boring you. So yeah, big ups to the organic growth of that whole community in that space, that safe space to partake or to just intermingle or learn from the different speakers that you guys provided. So big ups to you, Av, for you know, following through and getting that stuff done. I know it's no joke.
It just looks at the tagline there with conversations in Ken's Cafe. I think that's what we're trying to do with Grow Up. I think that's what we're trying to do with it. It's all about the biology as well, and here the soil matters. So yeah, it's about bringing people together, their minds, their ideas, and just continually spawning off that.
Yeah.
Well, like after our show today, we got on with a young lady that's developing some testing equipment that looks quite promising for a lot. Us doing our own testing at a price point that most people will be able to afford. So that's going to be coming up soon as well. But we've got Grow Up again in what is it, September?
October 1st and 3rd in Victoria.
I'm
looking forward to seeing how that rolls out.
And have we got Leighton booked? I think for I think he was talking to Shay LeBlanc from Rosebud Farms about the Unicorn Cup. Did he confirm that with you, Leighton? Has he lassoed you?
I don't believe he has as of this point. Where is that? Where's the unicorn?
That's in the southern part of BC and central BC. It's just across the border from Washington State in central BC. So I don't know. I'm hoping that he can lasso you into coming up. I know we had Kevin Jodry there last year, but we'll have to see.
Yeah, you know, I'll be honest, that little 3-day jaunt really did kick me in the you know where. I mean, I was pretty damn tired, and you know, getting up at the ass crack of dawn just throws my circadian friggin' rhythm completely out of whack.
Yeah.
So I will travel when necessary. I know Poe he settled in with his trainer, so he was okay. But boy, did he was happy to see me. I mean, he damn near freaked out, had a heart attack. So it's not fair to him, you know, if I do a ton of traveling. But if you know, if the need arises, you guys know how much I believe in education, in promoting, you know, this plant and all plants and all things healthy, living soil biology, etc. So I will make sacrifices as necessary.
And Poe would be able to come to that one. That one's dog friendly.
Well, the other thing though is to get him. He's too big to sit in my lap on the airplane. Well, he's not too big to sit on my lap. He's too big to put underneath in a one of those dog carriers. Plus, he would not go in a carrier. He had a really bad experience at a very young age with a crate. Pauline was trying to crate train him, and he just lost it. So I would never put him in a box. And I don't know as I can bring him like on a plane without being in a cage, right? I don't think they allow that. So, and the other side of that coin is that I could drive there. I've done Bellingham to Oxnard a number of times. It takes 2 days, and I burn a fuck ton of gas. At least a couple $100 of gas, a couple hundred hotels to get up and back. So it's it's more expensive to do it that way. I mean, I thought about doing that, going up to Grow Up, but I looked at it was like 1,200 miles. It would have been 4 hard days on the road up and 4 hard days on the road back, and it was just like, there's no way I can commit that time. So, but I
need me to come down in the motorhome and grab you and bring you back up, then you're not driving.
All right, there you go. Now you're talking, because you could sleep and drive, and you take shifts and get it done.
Yeah. So do you have anything coming up in California that you're going to be at? It's like, I know last year you were doing the competition for,
So what happened is we almost had the compost cup again this year. We were going to do it at Rodale's Field Day. And then there's this heirloom seed festival that's going on in Ventura in the fall and September. We're looking at working with them. There has been some communications back and forth. And then there's another event, the county fair, who also we've entertained or begun a conversation with them. So hopefully, one of these people get excited enough to say, okay, let's do this. And I think that we already have the website in place. We have the communications director. We've got all the judges. So it wouldn't be hard to put it together, put it that way. So hopefully that happens. Another interesting thing was that I've got a meeting with a screenwriter, a cannabis grower, compost maker, who are all very interested. And the cannabis grower has an in with some producers to produce a version of Kiss the Ground, but instead of just pointing out problems, putting on solutions, because that's the biggest problem we have right now. So hopefully that meeting goes well on Thursday, and we give him enough information to pitch to producers. Hopefully the producers are open-minded enough or have at least begun to talk regenerative organic. And if that's the case and we pitch it correctly, then I don't see that as not happening. It'll probably take a couple of years. I don't know how long these things take, but I know the one that Kevin did on the vineyard versus the cannabis grower was at least 2 years in production before it actually hit the theaters. But so we'll see. I mean, it's fun, exciting stuff.
Cool.
And so that's pretty much it as far as that outside of the house, so to say. Abby, you have anything else coming up soon?
No, just a little bit more conference prep. Trying to figure out what to talk, who to bring to MJ Biz in sort of end of November, December in Las Vegas, and before that the Grow Up conference in Victoria. So I'd like to see a little bit of an emphasis around bringing cannabis back as medicine, and so hopefully we can have more discussions around that. And other than that, I think from the conference, just trying to follow up with a lot of those growers and see where they're at implementing the ideas that came across. So
do you have your fingers in MJ Biz, Av?
I'm on their scientific advisory committee.
Oh, nice. Cool.
So that is definitely one of the things that came across at Grow Up is just the fact that I think they're getting those people in the same room and having that many minds together and just bouncing those ideas off. One of the common labs that we use in Canada, to whether it's for tissue analysis or for soil, is A&L Labs. Comparing that, and they use a a slightly different technique. It's not a malic, but it's not a saturated paste. They got something sort of halfway in between, and then trying to compare that to a Logan's lab. And so for those who are, whether they're designing their own soils or using living soil bed systems or horizontal bed systems, trying to get a really good understanding of what's going on in those soil tests. Yeah, that's a real challenge. And maybe that's a topic that we can bring up and perhaps bring in folks who are more experienced at interpreting those tests to talk about them. Yeah.
So I have some important information that I need to tell you, Av. So as soon as we go off the air, just stay on for a couple minutes, okay? I don't feel comfortable saying it publicly right now.
And that's the other one that you brought up. One of the points that you brought up in our regenerative ag session, just the idea of folks having aquatic systems on their land. Or somehow incorporated into their gardens. I'd love to have another conversation around that. Once again, one of the champions in the Cannabis Cup, and two of the entries in the Cannabis Cup were in fact aquaponic cannabis. I'd love to have a couple more aquaponic folks on just to talk about what's going on in that living water as well. And so,
all right, I'll reach out to Charlie and Eric, because both of they are very much deep down that rabbit hole, and they're not only teachers of it, but they have a tremendous amount of experience working in that industry and teachers. So they're not. This is an anecdotal. It's more of a professional environment. So I'll definitely reach out to them. And you're right. We do need to have more conversations about living water as well as living soils.
Yeah. Yeah, what Gerald was talking about. And Darker says we need cannabinoids and terpenes, how they've changed over time.
That, when you get into these secondary, tertiary metabolites, it's complex to say the least. The testing, we're actually, Ken, I pitched this morning to an investor for Ecosphere. We are actually already going down primary, secondary, and tertiary metabolites. I got Craig Tester on board. He's got another person coming on board. We're having a meeting tomorrow. We've already created the compass, and now we got to create the roadmap so that we're not wasting money doing testing that we don't need. But this is going to be something that once we open this up, then anybody can use AI or ChatGPT, any of these things to actually start to understand what is really happening based on data that we finally have for the first time in this community, in the cannabis community, because we do have, what was present in these different cultivars at harvest. So then it's just a matter of like sussing out, well, how did this breed mix with that breed, and what were the cannabinoids? And all right, let's take those even further and send them in for metabolite testing to understand, just exactly what James is doing. It's going down a rabbit hole that nobody's explored. No one's even stuck their toe in that water yet.
I love having the guys on that are writing the books, because that's what we have with James and a lot of the people, Gerald Pollock on water. They're writing the book on this stuff. They're at the cutting edge, and I'd love to get them together as a big group and just let them hash it out. The brain power would be just like, I just sit here and just nod my head going, wow.
Melt.
Yep, definitely.
Like a candle, you just be going
and quickly, I think, Yeah. Okay, guys. Well, as for me, I've got a couple of shows. I'm actually being interviewed tomorrow on Caribou Heart TV.
Congratulations.
Thank you. Then, of course, on Wednesday, we've got Andy Lopez on Gardens Untold Story at 6 p.m. Pacific. Thursday, we have a young lady, Opal, who is going to be coming on Conversations, or actually The Passion Within, and she runs her own apothecary. And we're going to talk about a lot of the things on plant medicine on that show. So check those out, guys. But other than that, if there's nothing else, guys, I think we're going to cut it there.
All right, and stay on for a minute, Al.
Sure, thanks.
Thanks, everybody, for listening. Hope you enjoyed
today, guys.